take a look at a case study where we'll be able to understand it more about how we can apply these concepts in practical purposes. Design of experiment is an important branch in applied statistics where your main intention is to find out what is the cause and effect relationships based on what are the different factors and the output of the process you are intending to have under some controlled conditions. So there are three basic principles which are used in design of experiments. First is randomization. So randomization is a cornerstone of statistical methods, and its main function is to reduce the bias. Application is useful if you want to estimate what is the experimental error which creeps in your process. But there is a downside of introducing replication in your process. So as you increase the replication, your cost can potentially go higher and higher with that. Employing randomization and replication, there is some extra source of variation which is often found to be present in the experiment. So experimenters often prefer to use local control also. So one of the techniques used for that is blocking. Now, what you do in blocking is you group some homogeneous experimental units based on some criteria. So ANOVA is used as a mechanism if you are predicting variables, that is the independent variables are categorical, and the response variable, that is the dependent variables, are numerical. So it is an extension of one sample t-test as a method to compare means of treatments of multiple populations. So this is what a generic ANOVA table looks like. First, check how many samples do you have. So if you have one sample, go for one sample t-test. If you have two samples, check if they are independent or dependent samples. Based on that, you can use two sample t-test or paired t-test. And when you have multiple samples, that's where ANOVA comes in picture. So if you have independent samples, then you use one-way ANOVA, two-way ANOVA, or factorial ANOVA based on the number of independent variables you have. And if you have dependent uh, populations, then you go for repeated measure ANOVA. Let's take a look at a case study where we'll be able to understand it more about how we can apply these concepts in practical purposes. There are four main stages in injection molding process, that is clamping, injection, cooling, and ejection. So first, in the clamping process, this is how the mold usually looks like. So first, in the clamping process, this is your mold. The mold closes, and the plastic pellets, which are melted at a very high temperature, are injected inside a screw-like plunger. This plunger is forced ahead, and it injects the molten plastic inside the mold. As the plastic is getting injected, the air inside it gets ejected through the tubes which are present, and until it is entirely filled with plastic. So after that, the mold is held in equilibrium till the plastic inside it cools down in the shape of the product. And in the meantime, the plunger retracts in its original position to get filled with the next batch of plastic. As the plastic mold inside cools down, the opens again, and the part which is formed inside now falls down and is caught inside beneath the mold with inside a bin. The mold closes again, and the next production cycle begins.